Ever picture a surgeon working deep inside the brain, navigating tiny, critical spaces?
It's pretty incredible stuff.
It really is. Today, we're going to do a deep dive into one specific procedure, endoscopic third ventriculostomy, ETV for short.
Right, ETV.
And we're going to explore the vital anatomy, kind of like the internal GPS a surgeon needs,
Right.
based on some detailed material we've looked at. The goal is to understand what they need to know without getting lost in every single detail.
Exactly. Think of ETV as basically creating a bypass, a new way for cerebrospinal fluid to drain when the usual paths are blocked.
Okay, a bypass.
Yeah, and to do that safely, the surgeon absolutely needs a crystal clear picture of what they're seeing through that tiny endoscope.
Okay, so let's start that journey. They enter the third ventricle. What's the first thing that visually jumps out?
Well, the sources highlight the mammillary bodies first. You'd see these two small, quite bright, white-yellowish rounded shapes.
Like yellow bumps.
They're a key initial landmark, sort of like the "you are here" marker on a map.
Got it, orientation points. So from there, where do they point the scope? What's next?
So from those mammillary bodies, the scope moves forward and a bit upwards. They're looking for the infundibular recess.
Infundibular recess, and what does that look like?
It's described as small and pinkish red, and that color is actually significant.
Oh, why is that?
It tells the surgeon there's a rich blood supply there from the hypothalamic portal system. It's a visual cue about the vascularity.
Ah, so the color itself is important information. Okay, leads us to the main event, right? The tuber cinereum, that's the target for making the opening.
That's the spot. The tuber cinereum. It sits roughly between the mammillary bodies, those first bumps we saw, and that pinkish infundibular recess.
And its appearance?
It's often described as triangular and, interestingly, frequently transparent or translucent.
Transparent, really. Within the brain.
Yeah, it sounds a bit strange, but especially in patients who have hydrocephalus, that fluid buildup this area can become very thin. So thin, in fact, that the surgeon might actually be able to see through it. They might glimpse structures underneath, like parts of the skull base, the dorsum sella, the clephas, even the basilar artery pulsing down there.
Wow, that is incredible. Like a tiny window.
Yeah.
Must be helpful for planning the opening.
Hugely helpful. Seeing those underlying structures gives crucial spatial awareness. But there's also something nearby you absolutely have to avoid.
Okay, what's that?
The arcuate nucleus. It's a critical part of the hypothalamus.
Hypothalamus.
Yeah.
Regulating hormones, body temp, all that.
Exactly. Loads of vital functions. The arcuate nucleus has this kind of horseshoe shape around the base of the infundibular stalk, right in that neighborhood.
So damaging it is a big no-no.
A very big no-no. You need to be incredibly precise to avoid it, and the other hypothalamic nuclei clustered nearby. That's why centering the opening is so paramount.
Okay, so precision is key.
Yeah.
How do they pinpoint the exact spot for that opening, the fenestration?
The ideal target, based on our info, is right on the tuber cinereum, specifically at the midpoint. Imagine a line between the infundibular recess and the point exactly between the two mammillary bodies.
Between them?
Yes, the intermammillary point. You aim for the middle of that line on the tuber cinereum itself.
And critically?
Critically, you need to be well away from the tip of that basilar artery you might see, and definitely clear of those vital hypothalamic nuclei on either side. It's a very specific, delicate target zone.
It really sounds like surgeons need this intricate 3D map etched into their minds. The material even mentions embryology. How does the brain's development play into this?
That's actually quite neat. The hypothalamus develops from a part of the embryonic brain called the diencephalon, and it forms bilaterally, meaning it's essentially mirrored on the left and right sides.
Oh, like two matched halves.
Pretty much. And because it has this bilateral organization, surgeons can make an opening precisely in the midline, right down the center, without necessarily damaging the core functional parts of those nuclei on either side.
So the midline approach works because of how it developed.
In essence, yes. Targeting the tuber cinereum right in that central spot leverages that bilateral structure. You're working in the space between these critical paired components.
Okay, let me try and recap then. For a successful ETV, the surgeon needs to first spot those bright mammillary bodies.
The starting point.
Then find the pinkish infundibular recess. Identify the tuber cinereum, which might even be see-through.
Right, that central triangular area.
And make the opening exactly at the midpoint between the recess and the spot between the mammillary bodies. all while dodging the basilar artery and, crucially, the arcuate nucleus and other hypothalamic bits nearby.
You've got it. And keeping in mind that whole bilateral structure of the hypothalamus, it's a complex interplay of visual landmarks and deep anatomical knowledge.
It really underscores that neurosurgery is just as much about profound anatomical understanding as it is about technical skill with the instruments.
Couldn't agree more. It's that detailed knowledge that guides every precise movement.
Thinking about this incredibly detailed landscape inside our heads and the skill needed to navigate it, it really gives you a new appreciation for the brain's complexity.
Absolutely. And here's something to maybe mull over. Think about other medical procedures, maybe ones that seem more routine from the outside. How many of those rely on a similar depth of unseen anatomical knowledge and pinpoint precision? It makes you wonder about the hidden expertise behind so much of healthcare, doesn't it?